amoeba. So, shockingly, I have some slides. So what I'd like to do today is talk about amoeboid lineages. Amoeba used to be known as the sarcodines or sarcodina. They were heterotrophic amoeba, were called sarcodina. Flagellates back then were called mastigophora. But these sarcodines turn out to be in 5 of the 6 major clades that we've been studying. In terms of the fossil record, testate amoeba are among the more famous fossils, and it's Susanna Porter and her team who worked on these beautiful scanning electron micrographs of shell-building amoeba from about 750 million years ago. So here's the fossil, and here are a couple examples of extant shell-building amoeba. What is an amoeba? Well, amoeba is a generic term for an organism that lacks pseudopods, or that lacks flagella, although they can have flagella sometimes, but they have pseudopods or false feet. These pseudopods are projections of cytoplasm that extend away from the cell body, and I'll show you that there are many different kinds of pseudopods. And not only are there amoeboid lineages, but some cells in multicelled organisms, like our own white blood cells and amoebocytes and sponges, are also amoeboid. What do the pseudopods do? Well, they're for flagella, they're for movement, like flagella and cilia are. And here is a movie of an amoeba using its pseudopods for phagocytosis. So there is a little ciliate. And these little ciliates are not winning, although you can also find images of other ciliates eating amoeba. But here, the pseudopods have now made a little corral, and I'm afraid that amoeba is not long for this world. So pseudopods are used for motion, and they can be used for feeding. The generic morphology of an amoeba, it has a cell membrane. They often have ectoplasm, a thin layer, and endoplasm, a thicker layer of cytoplasm. They can have vacuoles, crystal inclusions. What do? We don't know. They can have digestive vacuoles. They can have a nucleus. Here is an acanthamoeba, which is the organism that can damage your eyeballs if you don't clean your contact lenses. And here's the ectoplasm, the endoplasm, some crystalline inclusions, and here is the nucleus here. There are 4 main types of pseudopods. There are lobose pseudopods that are thick, finger-like in this polychaos. Here is some movement of a lobose pseudopod. This is how we imagine them. A lot of cytoplasmic movement and streaming is studied in these amoeboid lineages. But they also can be seen on their side. Dan Lahr, who was a PhD student with me some years ago, he made this movie by turning a camera on its side, and this is the same kind of amoeba that we saw before, but you can see its pseudopods interacting now with the glass slide it's on, and this is an actin-based motility mechanism, so quite different from the side. And amoeba, these pseudopods can just be beautiful. So this is a difflusia, a shell-building testate amoeba, and it is agglutinated. And here's, that one had glued stuff to its shell, and here's another naked amoeba moving. Another kind of amoeba pseudopod that we see are phylopseudopodia, and we're going to see these in a little bit in the rhizaria. But here is a short video of an amoeba with these fine pseudopods. So this one happens to fall in the rhizaria. Most of the the fine pseudopod amoeba, fall within rhizaria. And we do see these in labs sometimes. The third kind are these actinopodia, and I think a group presented on these for major clades. These are the heliozoans, like actinopheres. They have stiff, long pseudopods. They look beautiful. So again, heliozoa, sun animals. But it turns out these pseudopods that they have are incredibly efficient for predation. So these things will eat other organisms that are quite a bit larger than they are. And then the fourth kind of pseudopod are reticulopods, and we see these networks in things like forams, and also in an organism that we actually named called Corallomixa. So here is a movie of Corallomixa. I like to describe Corallomixa as an amoeba on your windshield. It's completely made up of this reticular network. So this is the amoeba. It has no major cell body, and it is capturing bacteria in here in these pseudopods. So the four main types: lobose, phylos, actino, and reticulo. And this was the basis for the classification of Sarcodyna. But of course, molecular data challenge this, and we'll be talking about this in class. Instead of going through the groups one by one, I just want to talk a little bit about forams because they're cool, and a couple groups in lab are working on them. Forams have these thread-like anastomosing, which means they can cross pseudopods, and they can bidirectionally stream. They have crazy life cycles, which I'll tell you a little bit about. Most have a shell, and they can be teeny to several centimeters. So in the fossil record, there are 10 centimeter long, that's about what, 5 or 6 inches, amoeba forams. And today in the deep sea floor is covered with 2 or 3 inch wide forams, all one cell, and they're down there in the deep sea floor feeding on deep sea snow and other particles. In fact, we don't understand their ecology well. Here's a naked foram, and this is a more traditional shelled foram. There are 4 kinds of forams. There can be naked, leathery shells, agglutinated, these glue shells, and then these calcareous. This is somebody's hand, so these might be 1/2 inch across. Not surprisingly, these aren't monophyletic groups. Here's just one example, Allogromius, which is one with a leathery shell, and we've had this in lab a couple of times, but it's really hard to keep in culture, and I call them the Spider-Mans of the natural world. They have these incredibly aggressive, effective pseudopod nets that they reach out of their shells in order to feed. So we're looking at a film of that pseudopodial network. Forams are found everywhere. They are predominantly marine, but there's growing literature on them in freshwater, soil, leaf litter. Here's a 1879 drawing of one, Gromia. In the ocean, they can be benthic, living in the sands, or they can be planktonic, floating in the water column. And what do forams eat? Everything. So they eat anything and everything. So here is a foram eating a copepod. They range from passive osmotrophs to ravenous carnivores. They eat bacteria, they eat other protists, including each other, and they eat small animals. This is a beautiful foram from Antarctica called Nodrodendoides, which can be about an inch tall. Here it is again, and this is taken in Antarctica, and I'm going to end with some pictures of Antarctica where foram diversity is greatest. So here's a small shrimp-like organism that this foram is eating for dinner. So in the case of these forams, the lab group brought some up for a brief period, and they put in some larger and larger animals. So they put in a shrimp that was 5 times the size of the foram, and you guessed it, the foram won, and what it did is it stuck its pseudopods up the nose and in the ear and in the mouth and ripped the inside of this animal out. So forams are really, truly voracious predators. Forams can also act as farmers. They can farm dinoflagellates, diatoms, all these different things. And what do I mean by that? Here's a classic foram picture. It's planktonic. It has very spines or pseudopods, depending on the species, that it can stick out. And during the day, it puts out these algae like a little greenhouse, holds them out into the sun, maximizes the capture, and then feeds on the photosynthate. There are also some that are kleptoplastidic, where they ingest the algae and steal only the chloroplasts. But these are well-established relationships, forams as farmers. In some of these cases, they have these spines that they'll put their algae out up on the spines and float through the water column. Here's another close-up. I think these are dinoflagellates sitting out on these spines. Forams have incredibly complicated life cycles. So here's one of this Allogromia I mentioned before with its leathery test. Here's the full life cycle. I would never want you to memorize it, just see it's complicated. They can be a single cell with a giant nucleus, and this guy can chop up into single cells with many nuclei, and then these guys can go through divisions to become single cells with one nucleus. So these are different stages, asexual, one cell, one nucleus, one cell, many nuclei. When they go through gametogenesis, it's even more complex. But what I want to tell you about is a project that Laura Parfrey did in my lab, looking at what happens when these cells grow. They start small, they get big, and then they chop into little babies again. And what she did is she estimated the ploidy, the genome content, as the cells grow. And when she fed her cells algae, she found as the cell grew, the DNA content increased dramatically. Because we were doing PCR back then, she wanted to switch them from a eukaryotic alga to bacterium for food, and they still grew, but as they grew in size, they had a significant reduction in their DNA content. This is weird because forams eating just bacteria generate less DNA, showing genome content is driven by food source. So a summary of this here, this doesn't project super well. Here's a foram with one nucleus and another bacterial fed foram with one nucleus, bright nucleus, less bright nucleus. The idea is when they're fed algae, they give birth to more daughter cells, they make more DNA, and the bacterial fed give birth to fewer daughter cells. This is a hypothesis. We didn't test that. But the end story is that forams may increase their fitness when they have a better diet by increasing genome content. So how do forams, they have this craziness, how do they reset? They do a process called Zerfall, which is nuclear cleaning. Here's inside the nucleus, here's outside the nucleus. The best image I have for this is Foyn 1936, and we don't understand what happens, but when they're going to reset their genome, they sweep extra stuff out of their nucleus, including DNA. What's going on? We don't know, but it's very dynamic. So I want to end by just telling you that among the other weird things about foraminiferin is that unlike most organisms, they have the greatest diversity in the poles. And so this is my colleague and friend Sam Bowser and one of his workers, and he went down with Laura Parfrey, who was the PhD student. They're in Antarctica. Look at these boots. You have to leave your boots on a shelf at night in Antarctica or they will freeze. And they are drilling, so they go out onto the ice shelf here, and they're drilling these holes. They put dynamite in these holes, and then they blow up the dynamite. So they're at the base of one of the dry valleys. So this is ocean in a valley. They saw one penguin, or Laura saw one penguin, a lost penguin going the wrong way. So this is ocean, but quite solidly frozen, and they stayed in this place away from all of the other research sites, because they blow open these holes and then they shovel out the chunks of ice. Here's the drill bit that they use here. Unfortunately, in this experiment, they broke their drill bit. And then they literally go down and they can vacuum the forams off, or they can go diving. So obviously you have to be specially trained to go diving in Antarctica. It's quite cold. They go diving. They can take these vacuums down and they can bring the sands back up and sort them. And this is what we know. This is a foram on somebody's hand. Foram diversity, really the greatest down here. And I showed you that tree foram as an example. So that's all I have for you today for amoeboid lineages. And I look forward to talking more.